health crisis both in the United States and around the world. While major scientific advances have made it easier than ever to prevent and treat HIV, there remains no vaccine or cure, and tens of thousands of people continue to contract HIV every year. Furthermore, there are a few challenges when it comes to tackling HIV. One, too few people with HIV are aware of their infection, and two, many people with HIV do not receive proper treatment. In particular, we decided to tackle the second challenge of proper treatment, which my colleague will next elaborate upon.
Using current chemistry databases to analyze molecules related to the causation of HIV, we are able to use machine learning to find the best compounds that inhibit the spread of HIV in a person. With our hackathon project, we hope to not only aid current biochem researchers with finding the best HIV treatments, but also to spread awareness and empathy around the HIV epidemic.
Now, let's take a look specifically at our product. So first, we'll see that we accessed a database called the Chembl database, and this gave us target proteins for HIV. Target proteins are things that allow HIV to spread inside of a person. So the target proteins we identified as being relevant are ID 0, 4, and 6. Protease, CCR5, and CAT5, as these will now be referred to in the future. So we'll see here that we then took these three proteins and found different compounds that address these and make it so that way they cannot infect people. So all three of these we chose because they show different ways of, they are different mechanisms to get HIV inside of human cells. Now, next thing we'll look at is our data processing. So we indicated the bioactivity of various compounds at addressing these targets by looking at literature for each of these for each target, as shown here with these links, and we identified how active a protein was how active a compound was at addressing each protein. So we'll see here that we split things into active, inactive, and intermediate categories for each compound at addressing each target. Now, my colleague will present graphs regarding this data.
Regarding the graphs for each of the analyses that we conducted, we found that for the targets that there's an inactive and active and intermediate activities. We can clearly see that inactive and active classifications are more prominent than the intermediate, as done in our analysis. And you can see the three bioactivity classes.
Furthermore, we'll see Lipinski descriptors. Now, these will be necessary to generate machine learning data in the future. These are a few different various factors at describing how effective a drug is at being taken orally. So things like its molecular weight and water affinity. And these things will be used to generate machine learning features in the future. Now, moving on to looking at the actual machine learning.
Now, moving on, we will need to extract fingerprint data for each molecule in order for the computer to better understand the actual components of the structure of the molecule. Now, we first need to extract the data sets for each compound and have the pre-processed data, and then extract their molecule structure and convert it to a fingerprint, as shown here in a matrix form. Then we need to have the PIC50 in order to have the output, and then concatenate them together. And then moving on, we will need to then form regression models using the fingerprint data. So first, we use the fingerprint data for each one. We used the data for each molecule for CCR5, CAT5, and protease, first with CCR. And we use the lazy predict in order to find the best possible regression model. As you can see here, the random forest regressor has the best R-squared value and the least roots mean square error. And we can see that the pattern overall is very correlated for the observed and predicted. Moving on to CAT5, we can see it's similar, that the pattern is very similar. We see that also it changed to the lasso CV as the best regression model. And the data is very correlated here. And lastly, we have the protease fingerprints. And again, the random forest regressor is the best. And the data is again very correlated.
Now, in conclusion, we'll see that our solution accurately addresses HIV as well as